Hey guys, welcome back to Genetic News Now. We've been discussing a series of genetics topics, and today the series is titled DNA Debunked. When you think about DNA, you gotta think about nucleic acids. Nucleic acids, the components of nucleic acids are DNA, RNA, and phosphates. Nucleic acids contain two kinds of nitrogenous bases. Those bases are purines and pyrimidines. Purines are adenine and guanine, and pyrimidines are thymine, cytosine, and uracil, which is found in RNA. In all, a nucleotide is composed of a sugar, a nitrogenous base, and a phosphate. Now, these nucleotides that I was talking about, these nucleotides have perfect matches. You see these perfect matches in base pairing. Base pairing is defined by Watson and Crick, occurs between two nucleotides that enables the double helix to maintain a constant width. As well, this is in line with Chargaff's base composition rules. Hydrogen bonds hold together each base pair, however, they are not the primary force that stabilizes the double helix. Adenine pairs to thymine via two hydrogen bonds, and guanine pairs to cytosine via three hydrogen bonds. And again, remember when we're talking about RNA, the thymine is replaced by uracil. Phosphodiester bonds are the connecting factor of the sugar phosphate backbone. The 3' end of a nucleotide attaches to the 5' end of another nucleotide. Specifically, the phosphate group attaches to the third carbon of one nucleotide and the fifth carbon of the next nucleotide. Structures. However, the main point of variance is the C2 carbon. The deoxyribose sugar, a variant of the ribose sugar, has an H atom at the C2 carbon, while the ribose sugar has an OH group at the C2 carbon. Therefore, the C2 carbon of ribose in particular allows enzymes to distinguish between DNA and RNA. However, in respect to the carbons, the 1' carbon attaches to the respective nitrogenous base, and as stated earlier, the 5' carbon contains the phosphate, and the 3' carbon binds to the next 5' phosphate. This is the hydrophobic bonds at the center of the double helix. Flat base pairs lie perpendicular to the sugar-phosphate backbone. In particular, hydrogen bonding, which is a specific hydrophobic bond, provides specificity in the number of hydrogen bonds between each base pair. Adenine binds to thymine and has two hydrogen bonds, and cytosine binds with guanine. Major grooves occur when the sugar-phosphate backbone is far apart, while minor grooves occur when the sugar-phosphate backbone is close together. When the base pairs stack, the phosphodiester bond faces outward and the nitrogenous bases face inward. The major groove is preferable as the DNA sequence can be read without opening up the helix through breaking base pairs. The Watson and Crick model focuses on bDNA, which has a right-handed coil. To determine a coil, imagine yourself walking up a spiral staircase when you look at the DNA. If the handrail is on your left, then it is a left-handed coil. However, if the handrail is on your right, then it is a right-handed coil.